Hello everyone, I welcome you to the Breaking Science channel. Once upon a time, some clever scientists at the University of California, Irvine and the University of Pennsylvania had a brilliant idea to fight Alzheimer's and other bad brain diseases by putting new, healthy cells into the brain. But there was one big problem. The brain was being stubborn and wouldn't let the new cells in. The scientists were determined to find a solution. They learned that in the brain, there are cells called microglia that are like the bodyguards, always on the lookout for any disease that might try to sneak in. But sometimes, even the best bodyguards can become a little lazy and not do their job properly. And that's what the scientists discovered, that the microglia in the brains of people with neurodegenerative disorders were not doing their job properly. So, the scientists had a new plan. They would try to use the microglia themselves to fight the diseases. But how could they do that? Well, it turns out that the microglia need a special signal from a protein called CSF1R to stay alive. The scientists found a drug that could block this signal and kill the microglia, creating space in the brain for the new, healthy cells to be inserted. But there was a catch. If the scientists didn't stop the drug before putting the new cells in, it would also kill the new cells. And even if they did stop the drug, the old microglia would come back too quickly for the new cells to take their place. This was a real quandary. The scientists didn't give up, though. They used a fancy tool called CRISPR gene editing to make a small change in the new cells they wanted to insert. They changed one amino acid, called G795A, which made the new cells resistant to the drug that killed the old microglia. They then tested their idea by injecting the new cells into brain models and giving the drug to kill the old microglia. And guess what? It worked. The new cells were able to resist the drug and thrive, while the old microglia died off. The scientists were thrilled with their discovery. They think that this could lead to many new ways to treat neurodegenerative disorders, like Alzheimer's. They could replace the lazy microglia with new, healthy ones. Or they could make new microglia that can recognize when a disease is trying to invade and fight back with special proteins before it causes any harm. The scientists believe that treatments based on this new microglial method could be developed within the next decade. They're already planning their next steps, which include investigating how they can use this approach to target the brain plaques that cause Alzheimer's and other diseases like CRAB. In conclusion, the scientists have found a way to overcome the brain's resistance to new cells being inserted to treat neurodegenerative disorders. They use CRISPR gene editing to make a small change in the new cells, making them resistant to the drug that killed the old microglia. The scientists believe that this could lead to many new treatments, which could be developed within the next decade. And they're already planning their next steps to investigate how to use this approach to target diseases like Alzheimer's. Thank you very much for watching. Support the channel with a like and share the news with your friends. See you soon.